Welcome to the lecture series on food microbiology. So far in this series, we have discussed on what are the common microbes which are prevalent in different types of food, how we can prevent or reduce their presence in the food. When we do packaging, how do we reduce or how do we safeguard the food? And we also talked about the detection methods, especially for foodborne pathogens. Following this, we have been talking about the different kinds of foodborne infections or foodborne diseases. We said that it has been divided into foodborne infections, intoxications, and also that this can be the causative reason for these can be multiple, either the presence of the microbes or the presence of the microbial toxins. Even a small amount would be sufficient to cause the disease. In this series, today we are going to talk about two major foodborne infections, salmonellosis and listeriosis. Salmonellosis caused by salmonella, they are regarded as human pathogens, but each salmonella species, or what we call as the subspecies, the way they cause the disease, the manifestation characteristic of the disease, as well as the severity of the illness is different in each one of them. Typhoid fever is the most severe, which is caused by salmonella, and consequently this was the first salmonella infection to be reliably described, that yes, typhoid fever is caused by salmonella, and that the reason it was proved that it is because of the bacterium. And all salmonella are classified into species Salmonella enterica. There are 7 subspecies in this, and nearly all the human pathogens Salmonellae are grouped under Salmonella enterica, subspecies enterica. Salmonellae are further subclassified into more than 2000 zeroers, and this classification is based on the O and the H antigens. This we will be discussing later. When we take the typhoid salmonellosis, these are caused by the zeroers Salmonella typhi and Salmonella paratyphi. The subspecies are A, B and C. And how does a person get infected? They are taken up by orally, that is food or water, and then the invasion pathway is through the intestinal tract. So, the food or through contaminated food or water, the organism enters the human body and reaches the intestinal tract. From the intestinal tract, they will enter the lymphatic tissue. First, they spread lymphogenously, then hematogenously. So, the generalized septic clinical picture of salmonellosis now manifests once it has started spreading both heterogeneously and the lymphogenously. And here, human carriers are the only source of infection for this typhoid fever. Transmission is either by smear infection or it can be indirect by food and drinking water. And if you look into the therapy, anti-infective agents are required for therapy, which are normally ampicillin, cotrimoxanol or 4-quinolones. An active vaccine is available to protect against typhoid fever. In 1856, if you see the history of typhoid fever detection, and that Salmonella typhi is involved in causing this particular disease, the English physician William Budd, he concluded that each case of typhoid is epidemiologically linked to an earlier case. Okay, so one person gets it from another person. So, it is epidemiologically linked to another case, and a specific toxin is disseminated with the patient. So, a patient, when he passes stools, excretes out the toxin, and indirect or direct ingestion of this toxin causes the disease in another person. And he demonstrated that treating the excreta of victims with chlorinated lime, which is bleaching powder, reduced the incidence of typhoid, which meant that the organisms which were present in the excreta were killed. The typhoid bacillus was first observed by Ebert and Koch in 1880, and 4 years later they were observed it, but it was Gafke in 1884 who first succeeded in growing it in the lab. And the paratyphoid bacilli, they are responsible for clinically similar condition which is called as the paratyphoid fever. These was first isolated by Eckart and Benzaud in 1896. How do you distinguish the various species in this genus? For that we have a specific scheme which is called as the Kaufman-White serotyping scheme, and this scheme describes the organisms on the basis of the somatic, which is called as the O antigen, and the flagellar, which is the H antigen, and then you have the capsular antigen. So, 3 types of distinct antigens are present on Salmonella typhi: the somatic or the O antigens, the flagellar or the H antigens, and the capsular or the VI antigens. So, based on which is the type of antigen which is present, the subspecies is classified, and this capsular V antigens is possessed by Salmonella typhi, Salmonella dubin, and some strains of Salmonella paratyphi also. Enteric salmonellosis develop when the pathogens are taken up with food. So, this is the second case. First we were talking about typhoid fever. This is the second type of salmonellosis, which is enteric salmonellosis. Here the source of infection is livestock, and these relatively frequent infections remain restricted in the gastrointestinal tract. Treatment with anti-infective agents is necessary only in exceptional cases. And how does this picture clinically look like? The salmonella are classified either as a typhoid salmonellosis or enteric salmonellosis regarding the relevant clinical pictures and epidemiologies. Now, it is not really known why the typhoid salmonella cause only the systemic disease in humans, whereas the enteric salmonellosis normally occur in animals also, not alone humans but also in other animals. And the enteric salmonellosis is restricted only to the GI tract, whereas typhoid fever the whole person falls sick, and it is restricted only to humans. So, if we look into the manifestation of the typhoid salmonellosis, the first step is attachment of the salmonella to the cells of jejunum. These are called as the M cells. Then once the cells get attached, they invade by means of endocytosis. This is a normal process by which a cell engulfs a particular particle, or how it takes in a food, like your amoeba gulping in the food particle. So, normal endocytosis. Then it gets transferred, and then by exocytosis. Phagocytosis in the subserosa by the macrophages, and then from there it gets translocated to the mesenteric lymph nodes. The organism proliferates in the lymph nodes. Then from there it will get spread up into the lymphogenous and the hematogenous dissemination. The secondary foci would be the spleen, the liver, the bone marrow, the bile ducts, the skin, and the Peyer's patches. Now, the manifestation as illness happens with increase in temperature, fever rising through the first week from to 39, 40 to 41.8 degrees. Then the further symptoms of typhoid occur like stupor, leukopenia, bradycardia, the splenic swelling, abnormal roseola beginning in the 3rd week, diarrhea, sometimes intestinal bleeding also because of ulceration in the Peyer's patches. Now, let us see the manifestation of enteric salmonellosis. Here, the attachment is to the enterocytes in the ileum and colon. Remember, this is restricted only to the GI tract. So, attachment to the enterocytes in the ileum and the colon, then invasion of the mucosa induced by the invasion proteins on the surface of the salmonella cells. They persist in the epithelial cells, and they also persist in the macrophages also. Then these produce the salmonella enterotoxin and cause local inflammation. So, once the enterotoxin is getting secreted, the illness manifests suddenly as diarrhea and vomiting, accompanied in some cases by high fever. and without any specific therapy or medication, the symptoms will reduce after several days. And in cases of massive diarrhea, symptoms may be observed that result in the loss of water and electrolytes. So, how do we detect this? Detection of the pathogen in the culture. You take out stools, get swabs, and then isolate the organisms, detect the pathogens in the culture. Selective indicator mediums are used to isolate the salmonella in the stool. Then identification of the serovars is done based on the metabolic patterns. Serovar classification is determined with specific antisera in slide agglutination test, and culture requires a minimum of 2 days to get the result. The typhoid salmonellosis can be diagnosed indirectly by measuring the titer of agglutinating antibodies to O and H. This is popularly known as the Weidel test. And to provide a conclusive proof, the titer must rise by at least fourfold from the blood sampled at the disease onset to a sample taken at least 1 week later. And typhoid salmonellosis must be treated with anti-infective agents, whereas the symptomatic treatment will suffice for enteric infections. And symptomatic treatment encompasses slowing down of the intestinal activity with loperamide, and then you replace the fluid and electrolyte losses orally with your electrolyte formula, which is 3.5 grams sodium chloride, 2.5 grams sodium bicarbonate, 1.5 grams of potassium chloride, and 20 grams of glucose per liter of water. And chronic carriers here are defined as convalescents who are still eliminating the pathogens 3 months after the end of the manifestation illness. So, sometimes the people will still carry these pathogens in their intestine. They would have fell ill with the disease and they would have got recovered, but even after 3 months, sometimes the parasites would still be present in them, the pathogens would still be present in them, and they would be passing it out with the stools. These are called as the chronic carriers. And how do these parasites sustain? In the scarified wall of the gallbladder. So, the chances of other people getting affected would be from the chronic carriers who still shed the pathogens after months after convalescence. And typhoid salmonellosis cases arises only sporadically or in the form of an epidemic because of a series of unfortunate circumstances where a carrier has been in touch or an infected person is in the food supply chain. Humans are the primary sources of infection here. By contrast, the enteric salmonellosis occur in the population both endemically and epidemically. Remember there, the livestock can also be affected in the case of enteric salmonellosis. So, they are the major or most important source of infection, and the pathogens get transmitted to humans by food. It can be by consuming an infected animal's meat or product or be in touch with an infected animal. And the main method of effective prevention is to avoid exposure, means clean drinking water, prevention of food contamination, avoidance of uncooked food in countries where salmonella occur frequently, disinfection of the excreta containing salmonella or from chronic carriers, etc. So, these steps will lessen the epidemic spread. And for travelers who go to typhoid fever endemic area, you have an oral vaccination, attenuated vaccination, which is called as a Virotif TY21A. So, this can also be a preventive, what we call as a prophylaxis against typhoid fever. Manifestation of enteric salmonellosis or typhoid fever, both are by the same organism, Salmonella typhi. The serovars vary, and accordingly, and for typhoid fever, humans are the source and humans are the only organisms which are infected, whereas for the case of the enteric fever, livestock can be infected, humans get the infection from the livestock, or it can be vice versa. Now, let us see the second type of foodborne infection, foodborne listeriosis. Listeria are gram-positive, non-spore forming and non-acid fast rods. And if you look into the relationship portal, they are closely related to Bacillus, Lactobacillus and Streptococcus. So, when the 16S ribosomal rRNA sequence data was analyzed, Listeria was placed closest to an organism which is called as Brocotrix, and along with Brocotrix and Listeria, together with Staphylococcus and Curtia, they happen to occur a position between the Bacillus group and the Lactobacillus group, and this occurs within the Clostridium Lactobacillus bacillus branch. So, it is closely related to these organisms. And the 6 species of Listeria are characterized by antigens that give rise to 17 zerovars. So, 6 species of Listeria are there and producing different types of antigens, and so far the antigens listed at kinds of 17 zerovars. And the primary pathogenic species is Listeria monocytogenes. It is represented by 13 zerovars. The most common zerovar among them is zerovar 1 and zerovar 4. Where do they occur? The Listeriae occur in nature. They can be found in decaying vegetation, in soil, in animal feces, sewage, silage and water. So, in general, if you look into the foodborne contamination aspect, it is seen in raw milk, soft cheese, frozen and fresh meat, poultry, seafood products and on fruits and vegetable products. So, any of these can be a source of infection to human beings. And the pathogenic and the virulence traits of Listeria monocytogenes produces beta hemolysis on blood agar, meaning they can destroy the red blood cells, and they can also produce acid from rhamnose, but not from xylose. So, this is a means of distinguishing between whether the strain of Listeria which is seen is pathogenic or not. If it produces beta hemolysis on blood agar and it also produces acid from rhamnose, the organism is pathogenic, it is virulent. And all the virulent strains of this organism produces a substance, a hemolysin, that is responsible for beta hemolysis on erythrocytes. And this hemolysin is also capable of destroying the phagocytes that engulf them. See, when a human body detects a pathogen, the phagocytes can go, the immune system's phagocytes can go and gulp them and destroy them. But here, the hemolysin which is secreted by the organism will destroy the phagocyte. And the substance which this hemolysin, the hemolysin which is secreted by Listeria, it is called as Listeriolysin O, LLO, and this Listeriolysin O has been shown to be homologous to the Streptolysin O, which is secreted by Streptococcus, and the Pneumolysin O, which is again secreted by Pneumococcus. And if you look into the mode of infection, see, animals, as I said, are the chief modes. From the animals, this can pass on to the environment, persist in soil, water, vegetables, fruits, silage, etc., etc. From this contaminated soil and water, it can still go on to the animal, and then the cycle will continue. And from the contaminated animal, if you happen to consume the product of the animal like milk, meat, carcass, dairy products from this animal, humans can get infected. Or from the environment, the plant can drink contaminated water or consume food which has been contaminated with the organism and get the disease. This disease again would now persist in the environment. The man will again excrete the organisms, which will add up to the environmental level of the organism's concern. So, this is a cycle which continues. The Listeriolysin which is secreted by the organism has a molecular weight of 6000 Daltons, and it is made up of 504 amino acids. And when does the organism secrete this? At the exponential phase of the growth, with maximum rate up to 8 to 10 hours after the hours of growth. And when the organism is consumed orally, it first colonizes the intestine, the human intestine. How this colonization happens is something which is really not known. So, once the organism has settled itself in the intestinal tract, it will now, from the intestinal tract, it will go into the other tissues. And this tissue penetration, normally it is, especially if it is a woman who has been infected and she is pregnant, it will pass through the placenta into the uterus. The organism will invade the tissues, including the placenta in the pregnant woman. Then it will enter the bloodstream, and through the bloodstream it will reach the other susceptible body parts. So, it is an intracellular pathogen. So, since it is an intracellular pathogen, it has to first reach the susceptible cells, and once it reaches the susceptible cells, inside these cells it has to replicate. So, get into the intestine, establish itself, then move into susceptible cells. Once inside the susceptible cells, start replicating. So, if it enters in the case of phagocytes, the entry is from two steps: directly into the phagosomes and from the phagosomes to the phagocyte cytoplasm. In non-phagocytic cell lines, the uptake of the organism requires surface bound proteins of the bacterium, and these have been designated as INI-A and INI-B, and the molecular weight of these proteins is 88 kilodalton and 65 kilodalton. Now, these proteins help in the entry of the Listeria monocytogenes into the host cells. The first protein is called as internalin, and on the mammalian cell it adheres or fixes itself or attaches itself to a receptor which is called as the E-cadherin. Then you have another associated protein which is P60. This is secreted by all species of Listeria. So, once the Listeria has entered the macrophages, it will escape the phagolysosomal membrane. See, normally the phagosome will unite with the lysosome, and the enzymes which are present in the lysosome will destruct the organism. But here the Listeria monocytogenesis survives inside the macrophage by escaping from the phagolysosomal membrane into the cytosol, and this escape is actually helped by the listeriolysin LLO. And once inside the cytosol, the surface protein helps in the formation of the actin tails, which will propel or push the organism towards the cytoplasmic membrane. And once it reaches the cytoplasmic membrane, around it a double membrane vacuole will develop. So, you have an LLO, you have two bacterial phospholipases, the bacteria get freed, and then the whole process gets repeated with the bacteria entering the adjacent cell. Again, evade the phagolysosomal action, go form a vacuole at the double membrane, escape. So, this process will continue. There is another Listeria species which is called as Ivanovi, which is infectious for sheep, and there it causes abortions, and it is also supposed to be a prolific producer of hemolysin on the sheep erythrocytes. The listeriolysin secreted by Listeria ivanovi is similar to the listeriolysin which is the human pathogen produces. It also is similar to, it produces an LLO like hemolysin, a sphingomyelinase and a lecithinase. Let us see what these things do in sheep. When humans get infected with listeriosis, they do not have unique set of symptoms, because the onset of the disease depends upon the state of the host or the immune system of the host. If it is a healthy person, or if the woman who is not pregnant, so non-pregnant healthy individuals who are not immunosuppressed, they are highly resistant to infection. So, even if by chance the organism enters, they won't get infected. So, the following conditions are known to predispose the adults getting listeriosis. Number one is neoplasm, second is an onset of AIDS, alcoholism, diabetes, especially type 1, cardiovascular disease, renal transplant, or if the person is undergoing corticosteroid therapy. If any of these conditions persist in a human being, then they would have a chance of the manifestation of listeriosis. But if you are a healthy person, then you will escape the infection. And when susceptible adults contract the disease, normally meningitis and sepsis are the common recognized symptoms for listeriosis. And depending upon the mode of infection, there are 4 distinct types of clinical symptoms. Infection in pregnancy. So, listeria can proliferate asymptomatically in the vagina and the uterus, and if the mother becomes symptomatic, it will be in the 3rd trimester. And symptoms include myalgia, headache, miscarriage, stillbirth, preterm labor, and the symptoms last for 7 to 10 days. Neonatal infection. Here also there are 2 forms. One, and early onset sepsis with listeria acquired in utero. So this will result. So if the mother was infected and the symptoms manifested in the third trimester, the child would be born preterm, premature birth. Then the listeria can be isolated in the placenta of the blood, meconium, nose, ears, throat. Another late onset meningitis, it is acquired through vaginal transmission. So the child is not contracting the disease in utero, but while it is being born, vaginal transmission. And also sometimes it is reported in cesarean deliveries also. So two cases, early onset or the late onset as neonatal infection. The third type of infection which is prevalent with listeria is central nervous system or the CNS infection. The listeria has a liking or an affinity for the brain parenchyma cells, especially the cells in the brain stem and in the meninges. And once it infects there, it causes encephalitis, meningitis and abscesses. So when the brain cells are affected, the mental status changes are very common, and then neuronal activity gets affected. So seizures become very common when the central nervous system is attacked and infected by the listeria. The last case is the gastroenteritis type of infection, which is a foodborne diarrheal disease, which is typically non-invasive. In all the other three cases, it is invasive. Gastrointestinal manifestation is non-invasive. The incubation period is 21 days and the diarrhea manifestation would be anywhere from 1 to 3 days. So as a food industry person, or if you are going to talk about foodborne listeria monocytogenesis, we would be more concerned with gastroenteritis. But if you are taken from a medical point of view, listeria can cause, depending upon the mode of infection, 4 different types of manifestations. And in the case of the foodborne infection, patients present with fever, muscle aches, gastrointestinal nausea, headache, stiff neck, loss of balance, etc. So today we have discussed 2 major human disorders or human foodborne infections, one which is caused by salmonella, the other which is caused by listeriosis. As we discuss about the organisms concerned, salmonella has also multiple serovars. They are each characterized by different types of the flagellal antigens. As you know, you have 2 types of manifestations. One is the typhoid fever, the other one is the enteric salmonellosis. When we talk about listeria, here also you have serovars and you see 4 types of manifestations depending upon the infection type. So as a foodborne infection, if you are talking about, question number 1 would be, what would be the precautions to be taken to avoid listeriosis? Who would be the people who have to be extra careful, especially with regard to getting infected with listeria? The second question which comes into mind is, what possible repercussions will be there if a diabetic person is suffering from typhoid fever? Remember, typhoid fever spreads throughout. So then what would be the repercussion? What sort, especially with type 1 diabetes, what would be the precautions to be taken? What would be the sort of repercurrence in the metabolism that person would present if infected with typhoid fever? Thank you.